talk to his mind and flesh out this paper a little bit for us. Thanks, Riley.
Thank you, Karen. Thanks all of you for coming, and I apologize for suggesting that we all read a cell paper. These are notoriously detailed and complicated and tricky to get through, but they also are usually very complete in some circumstances, so there is reason for that. Now, before I actually go into discussing the paper myself, I figure we should probably let the authors discuss the paper for us in a brief 5 minutes.
This study fundamentally looks at the role of glia in brain function, but even more broadly in human evolution. We've been looking for some years at the differences between lower species and humans in glial morphology, glial architecture, glial physiology specifically as it pertains to neuronal communication.
The traditional role of glial cells is to make the environment perfect for synaptic transmission. So what we have noted during several more evolutionary studies is that glial cells expand both in complexity and size and diversity as brain function become more complex. So for example, the human astrocyte and astrocyte are the most numerous of glial cells, is 20-fold larger than astrocyte in the rodent brain, and they have 10-fold more processes, so they're very much increased in complexity. So that question, basically raises the question, have glial cells expanded in their function and their roles, and maybe they're more implicated in higher cognitive network function in human than they are in rodents.
So if we essentially replace the mouse glial component with their human counterpart, essentially neural processing and information flow within the mouse brain. And so that was the fundamental point of the experiment. And it was a way to really investigate what are the roles, very fundamentally, the roles of human glia in human evolution. The operational design was actually very straightforward. We chimerized, as we call this process, of injecting baby mice with human glial progenitors. And these are glial progenitors that are derived from tissue, sorted, purified, and then injected into neonatal mice. And basically we're outcompeting that mouse population with the human glial progenitor pool. As the mice grow up, the human progenitors take over, they expand differentially relative to the mouse pool, they outcompete the mouse pool, and you end up with, as the mouse matures, with a mouse brain where all the glial progenitors, literally all of the glial progenitors, become human. It's actually been extraordinary to us just how different these animals are. So first of all, anatomically, the human astrocytes retain essentially cell-autonomous differentiation, so that in the mouse context, these cells actually grow to the size of normal human astrocytes. The progenitor populations, the progenitors look like human glial progenitors. So we looked at a number of different indices of the function of these human glia in the mouse brain. We looked at the rate of spread of a intracellular calcium wave. We found that that was the same in the human cells in the mouse environment as in a normal human cortical tissue.
So what we first tested in these humanized chimeric mice is what everyone in the neuroscience field would think of as our model for learning, and that is long-term potentiation. And that is basically that we prepare a brain slice from the humanized chimeric mice, we stimulate them, and if we stimulate them very strong, but for a short time, we will see a potentiation of the electrical responses in the slice. So we did that, and we found that the humanized chimeric mice exhibited much stronger long-term potentiation than the mice that was raised in parallel, but didn't have implantation of human cells.
And that told us that these chimeras really had significantly different neurophysiology than normal mice. And on that basis, since LTP we look at as essentially a surrogate for learning in a neural system, we then went to actual behavioral assessments. We first looked at fear conditioning, then we looked at a variety of other behavioral tests. And the bottom line is that the animals that were chimerized with the human cells had much more rapid learning in terms of the electrophysiologic surrogate, long-term potentiation, learning in actual point of fact in the behavioral testing. And so here we were essentially changing, if you will, the functional capabilities of these mice by virtue of the human glial chimerization, telling us that human glia have a species-specific role in species-specific intellectual capability and cognitive processing capability that previously we'd suspected, but this is our first proof of the point.
All right. Thank you, Dr. Goldman and Dr. Rittergard. So I'm going to, for the course's presentation, I'm going to kind of review the points that are made in the paper, go over them kind of a second time for some of you, a third time for others of you. And then I'll move on to the framework that Hank Greeley and colleagues put together for evaluating the ethical conditions for a very similar procedure that was never actually done with creating a human neuron mouse. And after that, I'll put in my on where I think that framework leaves this particular paper. And at some point in time during this, everyone should interrupt me a whole lot so we can actually get to the point of this, which is a discussion. The overall context of the study, as Dr. Goldman and Rittergard spoke about in that brief presentation, is looking at the, quote, other half of the brain, which managed to get to the cover of Scientific American in 2004. So this is the difference between the glial cells, which make up some 90% of the brain, and the neurons, which get all of the press. So there's an asymmetric focus on neurons, which are looked at as these privileged computers. They're actually doing all the computation. When information is flowing through your brain, it's flowing through your neurons. The neurons recombine information to do interesting things. The neurons where the learning takes place. And the general notion is that glial cells are just there to fill space if you're being, giving them the absolute least amount of authority. They maintain the blood-brain barrier, so they keep the neurons alive. They're kind of like the support staff. Some of them go as far as to myelinate neurons to increase the reliability and the speed of transmission of information throughout the brain, the creation of white matter and whatnot. However, in the late 90s, it was shown that glial cells have their own slow sort of activity. And this is calcium signaling. So as opposed to having these huge changes in voltage, which you see in neurons, which allow for very, very fast signaling over a very long distance, instead in glial cells you have these changes in the intracellular calcium concentration. Furthermore, by looking at this calcium signaling, you can imagine the synapse, so the locus of communication between two different neurons is actually a tripartite entity. So there's the presynaptic neuron, the neuron that's sending the signal, there's a postsynaptic neuron, the neuron that's receiving the signal, and then the glia are also intimately involved in modulating the synapse, in providing it with relevant information from the surrounding area, and regulating how it changes over time, regulating plasticity. Additionally, glia have been found to emit what are referred to as gliotransmitters, so chemicals that are emitted from the glia that then interact with neurons in a way similar to a neurotransmitter. However, there's still a lot of debate over exactly what that glial calcium signal means. It's not every input to the calcium system then leads to gliotransmitters being sent out all over the place. It's a lot more complicated. There seems to be weird issues with compartmentalization, different parts of a glia sending this calcium signal out. So it's not well known. It's not like they're just big, slow, dumb neurons. However, the big interesting difference that Goldman and Rittergard were getting at is that if you look at the genetics, if you look at the genetic difference between a human and a mouse, if you look at a neuron, there's not really a huge difference there. Goldman, even in this, if it wasn't in that particular video, he was quoted online as saying that if you look at them under a scope, you can't tell the difference between a human neuron and a mouse neuron. However, the glia, if you look at primate glia versus looking at the glia of mice, there's a huge difference. Let's see what I have listed here. Well, I'll get to the exact differences in a second. Additionally, this is more of a exactly how much this actually weighs in on this debate. Einstein's brain, when it was dissected, was found to have an increased percentage of glia. So there's this general idea of like, well, maybe glia are the hidden architects of human intelligence, and perhaps we've been looking in the wrong spot all along. So hence, has science missed half the brain, as the cover of Scientific American, which was following up on a review article by Douglas Fields in the same year, if you want to read up on that. So that brings us to this paper. And I'll start going through the method briefly, hopefully looking at time here. So interesting to note, Goldman and Engraftment have patented this method. So you can't just all go off making your own human chimeric mice. You have to at least ask Goldman and Engraftment first.
Which you could, couldn't you? I mean, so long as you do it for research purposes.
You have to pay for it.
Well, you'd have to, yeah, you'd have to go through them for the actual. Well, I'm not an expert on patent law.
I'm pretty sure that patents do not apply to research scientists working in their labs. However, as soon as you would want to create a marketplace deliverable for what you're working on in your lab, then you're going to get into all kinds of patenting issues because you use their patented method to make your
Well, I mean, I think I don't know patent law either. I don't know if somebody else has better ideas on this, but I do know that like they built certain constructs that they use in like their lentiviral construct where they infected the cells and things like that. And that might be something that they don't willingly share with others, whether under pressure or not. I know like just some of us that have been in the lab have asked for materials from other laboratories to create things and they conveniently don't answer your email.
If they're funded by a U.S. grant and they're publishing in Cell, they sign a contract saying that they will share it. I mean, whether that happens or not is entirely different.
Yeah. So like there is a, especially if it's funded by NIH or something, you're supposed to, but whether or not they do that. So I don't know, there might be a limitation on what parts of their protocol you can follow.
Yeah, definitely have NIH funding. Interesting on the contract. So this might get into more of the repercussions of this, but overview the method. So tissue was obtained from aborted human fetuses. Abortuses 17 to 22 weeks, which is just towards the end of how long we can go before the abortion is legal in this country. And tissue was harvested from the ventricular and subventricular zones. Tissue was then dissociated, and then so these individual cells that are now no longer connected to each other, there's no more or the initial structure that was there, just the genetics in perfect spheres, as a physicist might aspire to model. These cells were then selected based off of certain markers that they were expressing, so that you could select just these glial precursors. And then there is a specific difference between this paper and previous papers where they were looking more for the oligodendrocyte precursors to work with the myelin. The focus was again on the astrocytes, which is a different type of glial cell that's involved more with regulation of the synapse and providing a nice happy environment of regulating the blood-brain barrier, as opposed to the oligodendrocytes, which are more concerned with the myelination, with ensuring the conduction velocity stays at a certain level, creating white matter, getting signals from point A to point B really fast. So these are the astrocytes, the more warm, fuzzy glial cells, I guess you could think of that were possibly involved with this strange computation that really hadn't been looked into. So this work started in 2004 with a paper, as I said, was focused on remyelination. So in this initial paper, the exact same method, however, they then implanted this tissue into a very specific type of mutant mouse called a shiver mouse, which lacked all that myelin. So they were able to completely replace the myelin of this rat by providing a new source, which were these human glial precursors. In this case, they switched over to a different type of mouse which lacked a fully functioning immune system, which meant that these mice could live for up to 20 months, I think is the longest they had a mouse in this experiment, without any sort of reaction to the cells that they had in the brains. As a result of that, the mice that they started out with at least were blind, which is an interesting repercussion that they don't, unless you read into the paper fairly deeply, they don't initially get into that. So again, the initial focus of this was just trying to find a new way to study human myelin originally, and then later study human astrocytes in a non-human. Study them in vivo, but not have to actually open up an undergrad in order to see them working in a lab. So furthermore, so these cells were first selected. I don't really have access to a mouse, so I'll just... There. All right, so were first selected using this magnetic automated cell sorting and then transfected with a virus which gave these human cells a gene that allowed them to express enhanced GFP, meaning that all of the human cells that go into these rats are all glowy. So they're fairly easy to spot, which makes things a little bit easier for doing any sort of imaging of them. Once these cells were planted into the, grafted into these mice, so these were 1-day-old mice, P1. The astrocytes then migrate into the developing cortex, so the outer layers of the brain, as well as the hippocampus. And then as the mice get older, by the time they're a year old, these astrocytes have also colonized the amygdala, which some of you will recall is associated with emotional memory, and thalamus, which can generally be described as this information gating structure in the core of the top part of the brain. Striatum, which is involved with saliency and novelty. Note that their language is a little bit confusing in terms of how many of these cells were in there. In the myelination papers, which were earlier, they completely, every oligodendrocyte, all the myelin in those rats was human-derived. In this, there's definitely still lots of rat astrocytes in there, so they haven't completely replaced the astrocytes with human astrocytes. But they don't really give a number on the percentage, which has kind of weird implications for laws that weren't passed.
Yeah, I was interested in that too, because at first I was really impressed to see that there was so much penetration of the expression, like over time and then in deeper structures. But then they kind of blow you away with these pretty pictures, and then that you're not sure, they don't do any kind of quantitative study to tell you to what degree the infection, the process, the expression was, or did I miss that?
Yeah, I also a decent amount, and I did not see anything that told you exactly what percentage of the astrocytes in these animals were human-derived.
Because I'm just curious, how many do you need then to have a...
Yeah, they do tell you how much they started with. They started with throwing 300,000 cells into these individual rats, which is, the rats have something like 75 million neurons in their brains. Oh, sorry, mice. Sorry, I work with former rats, so forgive me if occasionally, but everything in this paper was mice, and then abortuses. So perhaps the first really amazing result, this could have been a paper all by itself, this result, is the fact that despite that they took these human cells from human tissue and then put them in a mouse, they still ended up looking like human cells. Which, when I tell this to artists who work with scientists and they're always trying to problematize everything, it's like, oh, you can't assume that it's just going to work like that when you put it in a rat or when you put it in a mouse or something. The fact that it still turned out to look like a human, despite that it was in this mouse context, they didn't really believe me at first. So specifically, what does it mean to be a human astrocyte versus a mouse astrocyte? Well, the human astrocytes are, there's different types of astrocytes, many different types of astrocytes in humans. They're bigger than mouse astrocytes, so that bar chart H over there shows that the mice astrocytes are get that 60 micron diameter versus the human astrocytes are the same size as they are in the actual human, which is they have approximately 140 micron diameter. So they're much bigger, and here you can see that beautiful picture of the green human astrocytes sitting along with the red mouse astrocytes hovering around it. And then additionally at the bottom there you can see a process that's, I wouldn't call it an axon or a dendrite, but a process that's extending. So this kind of yellow and red bit is coming out of one of these human astrocytes, and you can see that it covers a really substantial distance. It actually goes between different layers of the cortex, and so you can see all these different, the white arrows are pointing to individual astrocytes that are derived from the rat tissue, and so it's covering this massive domain of several different rat astrocytes, possibly enabling them to connect to each other. And Here they also show that the human astrocytes connect directly to the mouse astrocytes, allowing for communication between these different cell types. So that in itself was amazing. Also, they showed that this changes long-term potentiation. I won't go into much detail on this, but the general idea is that it's a really low-level change that is related to changes that occur in your brain when you're learning something. It's been shown to occur in the hippocampus when you're learning spatial tasks. Here they showed not only does LTP still work, but it actually is enhanced in some ways. The quote, memory, unquote, of this LTP is longer in these human chimerized mice. And it's important to note that they compared this to both normal mice as well as mice that had grafts from just other mice. So it's not just the grafting, it's actually the fact that it's this human tissue is doing this.
But I think one point to note is that this is an interesting phenomenon. We use it a lot to correlate with memory, but this is a purely experimental phenomenon that you generate in the lab. So this is not something that we know for sure would happen in a person. But it is consistently correlated with behavioral improvement. So.
Yeah. So this experiment was done on slices of mouse brain. This was not done inside the actual mouse. So yeah, it works better. Then what really blows everyone away is the fact that not only are there these small cellular changes, which you might expect. You might expect that maybe there's some small, minuscule, meaningless changes in the way the cells behave within the brain, but there's also behavioral changes. So they did this whole range of different behavioral tasks. Auditory fear conditioning, where you pair an innocuous sound with a shock, which was great because these mice were blind, so it was a nice task where you could learn how to do something, but it required vision. So that's up there in 6A, and they showed that these chimerized mice learn faster, and not shown is the fact that they also hold this in their memory for longer than both the two controls I mentioned previously. There's also contextual fear conditioning, which I have to hand it to these guys. They got to this point and they said, we really want to be able to do spatial learning. We want to be able to show not only can they associate a sound with something bad, but that they can also associate a room with something bad. But these mice were blind. So they created an entirely different line of mice, starting with a different mutant that wasn't blind, replicated all the prior results in the paper on this entirely new line of mice, and then ran several other behavioral experiments, and found that, yes, they learned to associate these small chambers with bad things are going to happen by measuring the freezing behavior. So mice, when they become scared, will stop moving. They also found that these chimerized mice were better at determining which was the bad room and which was just a normal room. And then lastly, these mice were more adept at identifying a novel object and investigating a novel object. So one could have all sorts of fun narratives with that. What was not shown in the actual paper was a negative result, which is in the supplement, which shows that chimeric mice are just as sociable as the normal mice. So they measure that by seeing whether or not the mouse wants to go interact with a strange mouse or whether or not it wants to just stay in an empty room. And these new chimerized mice were just as likely to run into, and meet the stranger as the normal mice were. Additionally, these chimeric mice were just as likely to prefer a stranger versus hanging out with someone they had already met. So they just as much prefer social novelty. There was no difference seen there, which gets at some other issues that might pop up in here. So I suppose before I run off, any questions on just like the actual experiment itself, on the scientific context for what was going on, or yes?
Well, yeah, with this one, were they just running them on a battery at this point, or is there some reason to think that they would have been more social?
So they don't provide a reason precisely why they did this task. One could hypothesize several different reasons. I myself think I'm glad that they did the task, as I think a lot of questions that would be asked immediately, had a picture of Stuart Little at the beginning of this, have they bred super mice? Have they created mice with unique human capacities for language or communication or something like that? And they show, not that directly, but they show this sort of immediate, there's no obvious difference in how sociable they are. So it's not like we've enhanced, a human social capacity in these mice as much as we've just enhanced their ability to run through, figure out mazes, and to realize that they're about to get shocked.
I think also maybe it's trying to get at salience or emotionality since they're looking primarily at amygdala-dependent tasks. So, I mean, maybe this is a way to show that it's not just the valence of the situation they're being in, of their environment or their task. It's specific to learning.
And they're not just skittish, right? So I mean,
Yeah.
like they have increased fear response, or they're just freaked out about everything. So if you generally have a freaked out mouse, then you know this would be different. But he's all right. He learns fear learning better.
The social novelty task also requires social memory, so you might have predicted that they would be able to recognize the familiar mouse more efficiently if they
Well,
were.
they didn't test for that though.
Well, they had novel versus familiar, so
Oh, that's a good point.
like there are some manipulations you can do that disrupt that. Like they can no longer recognize a familiar conspecific.
Is that?
I thought it was vasopressin, but whatever. I'm a molecular
I
biologist.
think that I think for me, this brought up, kind of related to your super mouse idea, like, is there a cost, if you wanted to enhance an animal with this, if you wanted to call it enhancement, because I'm making them smarter and making more human-like, they perform better on these tests, what's the downside, what's the drawback? And so, would you create a certain kind of suffering with sociability, like a deficit there, which maybe you wouldn't want. And also they talk about pain, and I don't know if you were...
Yeah, so I wasn't going to go over those controls, but I can tell you briefly, in the settlement they look to make sure that the mice just don't feel pain more, they don't respond faster when they get shocked, that sort of stuff, and they find that these mice fall within the usual spectrum. So it's not like they're just always in more pain or something like that, thus they might be more skittish.
Right, because the doogie mice respond to pain more.
So yeah, do you want to elaborate on that?
Not really.
Oh.
Are people familiar with these doogie mice? They have increased cortex and they're smarter and they do things better, but because they have this better memory, they're slightly harder to handle because you inject a mouse one time and you come back and maybe you forgot. But these guys are like, oh no, you're not getting it again. And there's some real molecular bases for these things happening, but generally they're a more skittish mouse because of this enhanced memory.
Yeah, they have, they're genetically engineered, so they're a little different than this because they don't have a graft or anything, but they have increased expression of certain part of the glutamate receptor, and then they seem to have be more sensitive to pain, so in their perception of pain. So that was kind of, that in itself is a little bit controversial what they mean by that, because what they did was they injected an irritant under their skin and then saw how much they licked that, and so that could be related to a lot of things, but they said they were more sensitive to pain. So then the idea was like, oh, I could be enhanced, but then I'm also going to be more sensitive to pain, and what kind of life is that going to be? I could be smarter, but in more pain all the time. Oh, sorry.
So that makes me, and I don't know whether they did this, but like, is glutamate just affected in these mice? Because astrocytes and
Yes, they do have a mechanism. I didn't go over that too much, but they are fairly certain that the changes that they're seeing are due to an increased expression of tumor necrosis factor alpha, and they didn't.
I think it's facilitating Glial receptors.
Right. Yeah.
Which I couldn't remember if that was what they did for the doogie mice too, or if it's a different.
I think it's NR1. They enhance expression of NR1.
This is the doogie mice are NR2B.
Oh, okay.
Yeah. So this was tumor necrosis factor alpha to increase phosphorylation of certain sites of GLUR1, which should enhance synaptic plasticity or increase its amplitude or something like that. So the really cool thing, I didn't really go into the details of this, but they found that some of these behavioral tasks, they can actually knock out this enhanced performance by giving these mice a drug which blocks tumor necrosis factor alpha. So if you are worried about super mice, then you have this control.
So if you wouldn't even need to do this whole astrocytes, can you just do the tumor, inject that
The TNF alpha?
Could you just inject that chemical directly and then just have the same effects?
You could do that, but then you're losing whatever control the astrocyte has over whether or not that particular substance is being sent out or not. So that would be a more blunt way of doing it. Definitely, you could definitely test some interesting hypotheses that way. I forget if they had something like that in either the actual paper or the supplement. But no. All right. So to be clear, I'm pretty daggum sure that this work was completely justified. And I don't really know of another way that the authors could have investigated the particular questions that they really cared about here. But that being said, there are a handful of eyebrow raisers, which I think individually they're fairly small issues that are very easily just kind of looked over. But I think that when you start to have these things interact with each other, you start to get interesting things about what the next step is going to be, and of course the overall problem of incrementalism. Where do you say, we're not going to go there? We're going to go up to this point, but no further. So first off, the rhetoric. The first line in the introduction here talks about investigating the unique processing capabilities of the human brain. So granted, we could just say, well, they were just trying to use this to motivate their paper. They didn't really think they were talking about how communication popped out or something like that. They weren't really talking about those sorts of things, but it's just interesting that that's still part of the rhetoric. Methods, of course, is the fact that the tissue was derived from human abortuses. Results, the graph changes were changing behaviors that are associated with a negative affect. So you do start to get questions of suffering that pop up here. I don't think they're extreme problems like, oh my god, these mice should all be killed right now because they're suffering all the time. But it's just interesting that's the direction this is going, or at least that's part of that direction. And then lastly, the fact that the method for generating these animals is patented. The patent, I looked over it a bit, but then got scared. The general gist of it is they patented the mouse in the 2004 paper, which was defined by the number of these glial cells that had been replaced with human cells. So they were looking at, I think it was an animal who has 30% human glia is patented, but they don't actually prove that that's the animal they're dealing with in this particular paper. But the other clause within there is they also said they were talking about the method itself that was used to generate these animals. So those are kind of the four hot button issues. There has been some yes?
Can you, sorry to interrupt.
No, do it.
Can you get human glial progenitor cells from adults? Like if someone, say in a car accident, like would that be like a form of organ donation? I don't know really about stem cells.
Oh wait, are there even progenitor glial cells in adult humans?
Well, that's what
I know neurons don't get replaced as you age really, except in a few cases, but I wonder if glia get replaced.
Like in the subventricular zone or like in the hippocampus, that's what I'm wondering.
So they actually dissected out of the ventricular zone and the subventricular zone from these abortuses. I don't know if you could harvest from adults. That's a very interesting question. I imagine it might be a lot easier to get it from abortuses.
Yeah.
All their skull and everything.
Yeah, it'd be a difficult operation.
What about skin cells? Like,
Do IPS?
What about like making this adult cells back into embryonic cells and then cause them to differentiate?
IPS.
right, that's what I'm saying.
Yeah, I think that's an option. However, I think there's still a lot of questions on exactly what the difference is. I mean, I'm not in that area. I know some people who are working with that. I think there's still major differences that aren't known yet in terms of what the difference is between an IPS neuron and an actual
There are some
neuron.
troubles. I mean, they're more likely to have errors because they're older and become cancerous, and that's one of the things that people worry about in low yield. But I think we talked about this in class that you can get commercially available foreskin.
Yeah.
So like you can use that and try to derive that back into your stem cells. But that's interesting. You can Google that.
And there's companies out there that will sell you human neurons that are derived from IPS foreskin. The company's name is iCell. I tried to fight them on NDAs, it didn't work very well. So one of there's a couple of points that I wanted to add in terms of the ethics, and there's a couple of points that I think people have already made when discussing very similar issues about the ethics. John Moreno had a blog post on Slate recently. This paper just came out, in March. And one of the points he was making was that there really, doesn't seem to be a big uproar about this. And I think there are interesting questions just in the fact that people aren't really that concerned. Perhaps glia, either because they've been relegated to this, lower status than neurons, which are near sacred. Perhaps they're not viewed as being as human as neurons, despite the fact that this paper is actually demonstrating that they're more human. And what was this point that I wanted to make? Right, so John Moreno had, of course, at the point that one can only wonder what the response would have been if it had been published during the 2012 election season. I'm sure there's all sorts of people that would have gotten up in arms about this. The way I view this paper is, we're actually watching the incremental changes occur here. This isn't, obviously they didn't demonstrate that they could create a Stuart Little. So we're not looking yet, at the very least, about adding unique human capacities, despite the wording in the paper's introduction, to these mice. But perhaps we should view this as the start of something and figure out how to just deal with these sorts of small changes, figure out if they are worthy of changing the way that we're interacting with these mice, the way that we view these mice, how do we make those changes as we develop these new organisms. However, in terms of establishing, I was so happy when I found that picture. Image searched Hank Greeley, and it was like, picture of Hank Greeley. Apparently he got some sort of award for being like a noble or brave human or something like that, along with Barack Obama. And they made a cartoon of him.
Awesome.
So you too
That's
can
how
be
you
a
know
cartoon.
you've arrived.
You're a cartoon. So Greeley et al. 2007 in American Journal of Bioethics, which is Greeley, Cho, Hoggle, and Satz. They put out a paper on the human neuron mouse. So this was on experiments that have been proposed by Irving Weissman to do something very similar where you have a line of mice, the neurons all die within the mice, but then you replace the neurons with human neurons. And Irving Weissman went out and actually asked Hank Greeley, like, all right, I'm planning on doing this experiment. It's a little weird. Do you have any suggestions about how I should be going about navigating the ethics? And Dr. Greeley created an ad hoc committee. They talked about it for a couple of years. They gave him some suggestions. Weissman didn't end up doing the experiment for various reasons, but he's still kind of like, maybe in the future, it could be done. But even just this idea of creating this human neuron mouse created a decent amount of uproar, including George Bush in one of his addresses talking about we have to pass laws that will prevent hybrid human animals.
Mice overlords.
Yeah, then the internet, I'm sure, took that to mice overlords. And there was actually a law that was drafted that I think has been hanging around since 2005, still hasn't really actually seen any major discussion on the floor about preventing at least certain kinds of human-animal chimeras as a response to this, proposed study by Weissman indirectly. But Greeley et al., their actual framework for looking at this, they brought up what they thought of as like the 5 core issues. Just about everyone wants to focus on issue number 4. That's what a lot of the other literature prior to these guys has been focused on, and as well as afterwards. It was most of the responses to this article were focused on that point number 4. However,
Can I ask a question?
Yes.
I don't know what that means, conferring humanity.
Yes, neither do I. I'm going to go through, so go through the points. So one, sources of the brain tissue, the fact that these tissues are derived from, would probably be, the tissues that you would use for any of these sorts of experiments would probably be derived from human abortuses. The issue is that perhaps this would influence whether or not someone was going to get an abortion or not. The second was the potential for more pain and suffering in these mice, which gets along to what if we change the mice in some sort of way so that they can, either one, maybe this is just going to give them seizures. Like, the fact that this experiment that I'm showing here worked as well as it did is a near miracle. I definitely would have expected some sort of epilepsy or some sort of major problem to occur. So there's a good chance that any of these manipulations are going to cause more pain and suffering in the mice. A sub part of that question is whether or not we might enhance the moral standing of these animals and then not change the way we behave around them, not change the way we interact with them. So perhaps we've made it so now instead of thinking of it like a mouse, you have to think of it more like a rat perhaps, and now you have to give them a little bit more of an interesting world for them to engage with. Maybe you have to start thinking about psychological health at some level in addition to just physical health. And the idea was that if we did that, we probably wouldn't actually change the way we're behaving with these mice, and we'd later go back and be like, oh my god, we were torturing these new mice, but at the time, we wouldn't really bother with that.
Or that means you would have to change like the IACA.
Yeah, which I think that, and along those lines, I think that the first of these questions, which I think especially is most interesting is on number 2 here, is what should the technicians be doing? The people who are interacting with these mice on a day-to-day basis, should they be constantly evaluating the development of these mice? How do they create a new culture almost associated with these animals that have never existed before?
I just was going to ask, we don't really know what the reaction is going to be, though. I mean, you mentioned blindness that wasn't really discussed, and also, I mean, in the passage of time, you don't actually know what might develop.
Yeah, I agree. And the blindness was they didn't discuss it, but it was known beforehand just with that type of mice. But yeah, that's the issue. That's the scientific question, is you don't know how you're going to be changing these animals. I didn't mean to totally throw off your point. And so, yes, I agree there. We don't know what's going to change. Yeah, we'll just skip to
Has anything else surfaced?
In these mice?
Yes.
So, not that has been reported, at the very least. This paper is really the source of information on what sort of changes these mice have seen. I think it would be very interesting to have worked as a lab tech in that lab and have seen the development of those mice and have been the sort of person who was interacting with them enough that they might be able to detect small, subtle behavioral changes, small changes in interactions between them, which is, yes.
But to be clear, they didn't become blind as a result of this.
No. They did not become.
And so, in other words, we're already working with mice to whom significant insult has been committed in any conventional sense, right?
Certainly, yeah.
And so, in other words, this has to be analyzed in the context of what's considered to be acceptable with regard to treatment of laboratory animals more generally, right?
Certainly, yeah. Creating a line of mutant mice that every one of them is blind from birth. Yeah, what are the other limitations of that? And I think it's generally accepted that if they become a useful mouse for scientific purposes, then we're allowed to sacrifice the natural life of a mouse for a slightly different life that is now more useful for scientific purposes. I think that's generally accepted as long as you make the case in your IACOOK that there is a reason for doing this. Now the question gets to be then, if they are blind, does that mean you have to change their environments to prevent certain types of injuries that they might not have otherwise had? You have to change, I'll leave it at that.
Do you have more? I don't.
Yeah, I've got a little bit more. I also, sorry, I did not mean to throw you off like that. I meant to kind of go through a brief overview of these points, and then people did what I asked them to, which is to have questions. So yeah, risk of conferring humanity. The idea here is that you kind of this great fear of creating a super mouse. So creating a mouse that has unique human capacities. I think that Greeley et al. In their paper, it was very surface level. They didn't really go into exactly what they meant by that, but they definitely said learning and memory aren't in there. Learning and memory are something where, just because the mouse can remember something more doesn't change the way you think of it in terms of like, is it a human now? But communication, social ability, those sorts of issues, anything that looked like the development of language would certainly, and rational thought, those sorts of issues are what they at least meant by that. I think that...
But I have a question on that.
Yes.
So my understanding is that those things that we just describe or ascribe as uniquely human are actually not differences in kind from other animals, but differences in degree. And so if it's only a difference in degree, why should we throw the epithet unique before it.
Mm-hmm.
So the rhetoric that you had on a previous slide, that we're identifying what's unique about human brains, well, I'm sorry, it's not unique, it's just differences of degree in learning speed, learning potentiality, et cetera, and behavior patterns. So I'm still not clear about what it means about conferring humanity.
But Jonathan, you certainly know that there are people who don't adopt that difference in degree, that these are essentialists who adopt a difference in kind. And what they are profoundly concerned about, they're coming at this from an Aristotelian kind of perspective,
right,
where they're the animals and we're the humans, and there's this species to species transgression that's going on here, and that's where they're coming from. And I have a hunch that what they're really thinking of is that this sort of experiments desecrates the human, because we're taking what is this human, excellent substance and putting it in a non-human, and therefore it's a slur to human dignity.
And conversely, it's desecrating the animals.
Exactly.
These mice are perfect in and of themselves.
Right.
They're great mice. Why screw with them? Why try and create them?
But these conservatives would not be worried about the mice. Metaphysics, they would be worried about the
By
human.
the way, Jonathan, Dr. Frey puts on a conference that will be probably next year also, discussing a lot of these issues. So this year was called Beastly Morality.
Beastly Morality.
Do you have any of that information archived for people to?
It's hopefully coming out in the book.
Okay, so there you go.
So the Animal Ethics Conference organized here through the Center for Ethics, Dr. Craig leads that.
Can you really go back to number 3 and help explain how that's different than number 1?
Right, so that actually gets to the point that you were making in terms of what should we be doing with human tissue. So the propriety, use of human, stay awake. So one of the points that was brought up, you're taking what was human tissue, you're dissociating it, doing all sorts of changes to it, but then you're now including it in a mouse. So whether or not that is a proper use of human tissue, for instance, we don't just throw out dead bodies. We bury them. We have all this established protocol for how we deal with them. So that is one of the issues that gets to human dignity, which it also does pop up here. What was that?
Oh, just thank you. Okay.
Okay. So I think I had an additional point on. Nope, one button. Okay, proprietary use of human tissue. We agree. And then the last point that Glial et al. Look at is the risk of public support for science, and whether or not this could be some sort of, even if the scientists themselves don't think of this as being an issue, whether or not it would be understood to be a major issue, and thus there might be changes in who is going to be allowing funding to go to the NIH to fund this sort of nonsense. Although certainly in the case of this particular paper, that doesn't seem to be an issue. There doesn't really seem to be anyone who's just getting up in arms about the fact that we've desecrated our glia by putting them in these mice.
I will bet you that if George Bush or his ideological kind were still in office, and he still had the same kind of composition in his bioethics commission, they would be very, very upset with this kind of research, and probably take very forceful measures to cut off NIH funding for this kind of chimera.
It was 2007.
Right?
It was 2007.
Yeah, so this paper was definitely...
He was in office.
Well, so
that means he probably didn't know.
No, but this was
Oh, but it wasn't NIH funded.
This was hypothetical.
This was really looking at the ethics.
That's right, I beg your pardon.
As opposed to, this paper came out like a month ago.
Yeah.
So the Han et al. Forebrain Engraftment came out about a month ago.
But if these data were back then, if
Yeah.
these data had come out earlier.
It's not new for inbred strains to have things like blindness.
No.
Right? That's actually relatively standard.
Yeah.
Like Sprague Dolly rats are pretty much blind. And a lot of the mouse strains are deaf.
Yeah, so those sorts of changes, I agree, are definitely all over the place. The issues that, the reason I think this paper is especially interesting is, I might as well just go to this. The idea of changing the mouse's understanding of the world, not just its perception, not just tonic levels of background pain or something like that, but changing the mouse so that it's going from maybe a mouse to maybe, if you have some sort of weird hierarchy of being where you're allowed to torture this, you can't make this uncomfortable, and this you have to look at its psychological welfare, and the mice are going up that steadily.
Yeah,
perhaps.
I'm sure it would come, but I'm curious as to their propensity for anxiety, depression, and empathy.
Yeah, and honestly, I don't even really know enough to say what that looks like in a mouse. I mean, I'm still in the camp of people who are like, yeah, you're talking about autism in mice? What is this? That doesn't make any
Well, they
sense.
certainly have learned helplessness models for depression, right, and anxiety tests like the open field of disease. They're willing to go out in the middle of an arena.
And then there's some evidence that rats might be expressing empathy when they release cage mates who are trapped over a tree. So
Yeah, that's where I was thinking.
That's a very primitive kind of, I just wrote a paper on that. It's a very primitive kind of empathy. And then the thinking is that when that rat goes to comfort a cage mate, what the intention is really for the rat to relieve himself or herself of the discomfort that cage mate is experiencing. It's not real empathy where you take the perspective of another and
I'm not sure that that's entirely different from when I comfort
Yeah.
my child.
well,
When my 3-year-old is screaming and I grab him with Tic Tacs.
It's a primitive and absolutely necessary component of empathy. If you didn't have that discomfort being triggered in yourself, you wouldn't.
Right, but I think what we're establishing is that that doesn't make us any more advanced than them. Like, I don't think that what we do is that different from that. I mean,
Well, in a therapy session, it would be.
Right.
But I guess on a conceptual level, to start discussing that, I mean, that, whether you call it primitive or not, if you agree that it's a fundamental building block, then you stop being able to say these are only humanly privileged
Right.
traits.
Right.
So that's kind of what this is getting at when you're talking about getting more human. And maybe actually some of us are coming to understand more neuroscience that these traits aren't as privileged to humans as we thought. So this is actually kind of behind the times in that.
Not only is it not privileged with humans, but think it's a wonderful evolutionary artifact, right? I mean, the fact that when you see another of your conspecific suffering, for whatever reason, you go over to comfort it to try to help it out. I mean, that's an evolution. That's an adaptive plus.
And then there's evidence of trans species comforting that goes on as well, not just humans to other animals, but other species amongst themselves comfort each other. But this again testifies that what we do is just more or less different in degree than different in kind from what animals do and how they think. And so that point about conferring humanity doesn't make any sense in my mind. It makes more sense to say there's the risk of conferring personhood to it. Humanity refers to biology, and as these scientists are showing, biology can live if it's xenotransplanted. Wow, that is pretty fascinating science. The ethics of it is, so what? What do we confer upon these people? And personhood is the legal concept or the moral concept. And so that's what I'm wondering about, is how much brain matter, how much human DNA should we be pumping into these other species before we do confer upon them some semblance of personhood, where they are granted some, not just subjects of moral concern, but subjects, but they become moral agents in and of themselves.
And that, just quickly, that trend is, in animal law, is becoming, that topic is all really popular, and in the last, decade, in the US and Canada, we've gone from 10 schools that offer animal law to 120 or something like that. So it's becoming a really pressing issue. And it goes beyond actually animals to trees and all kinds of things like that. So there are a few precedents. We should probably just go answer the last few questions.
I just wanted to make an observation that might help disassociate questions about humanizing from the other ethical questions, and that is it's quite conceivable that the same effect could be achieved without transplantation of human glial cells, but by inducing the kind of change in indigenous mouse glial cells. And so certain ethical questions still survive. And I think it's important to distinguish the capacities and enhancements in behavior or flaws in behavior that the mouse acquires as a result of new types of glial cells from the origins of those glial cells.
Yeah, I mean, it's sort of on a similar vein, the difference between humanity and personhood. personhood and the sorts of changes we can induce in a mouse, like this has just made me think, okay, we took a human glial cell, which is very, very different from a mouse glial cell. We put it in the mouse, it retained its human-like properties, and of course it had some effect on the mouse brain. Like you made a huge manipulation of the mouse brain. It's really cool, but to me, like, the fact that it changed the mouse behavior appreciably is not too surprising. But then the real question becomes, like, if you were to take the same human-derived glial cell populations and put them in an animal that already has huge arborized astrocytes, like another great ape, right? Would that be humanizing a great ape? Would there be a change? Or would it be these types of astrocytes are pretty much interchangeable between great apes and it wouldn't actually affect the ape behavior? Or if you were to take astrocytes from an animal that's sort of in between, like, I don't know what a dog's astrocytes look like, but if there's somewhere in between, like, would you be able to detect a change in mouse behavior if you gave a dog astrocytes? How much of this is really humanizing the animal versus just taking a cell type that was never there before and really changing the way that these neural networks are constructed and potentiating LTP because you're making a really mechanical change to the brain. So I guess, like, I don't know if a neuroengineer can speak to that. Like, you're swapping out some of the parts, so so you're getting new properties out of this machine that.
It's like organ donation. It doesn't make you like, you know, Tom if you have Tom's liver.
But it is making like Tom if you have Tom's limbic system.
Mm-hmm. Yeah.
Yeah, and even on one level, yeah, it certainly doesn't make you like Tom in a human sense because, yeah, your liver isn't really used for creating what we thought was Tom unless he just really was good at digesting alcohol or something. On the other hand, I mean, there's still fiction, though, that uses organ donation as a key point. I think there's a really crappy Clint Eastwood
comedy about it.
Yeah, it's a Clint Eastwood gets the heart of, you know, somebody's sister, and then they hook up. So, you know, what is this kind of weird relationship that, you know, on a level, like, you certainly wouldn't think that Clint Eastwood is this person's sister, but at the same time, there's that fact that's always in the back of your head, and it's definitely influencing the way that you're perceiving the situation. Whether or not that should be something that influences the law, you know, probably not, but I think it still changes the ethics. It still changes the way that people look at that relationship.
I was just going to make a comment and to say thank you. I really appreciate, I think you highlighted what's the really interesting ethical question here. Because, I mean, clearly it's not the mice aren't approaching something that we would consider, they're not going to confer human personhood on them. Use personhood, not humanity, sorry, personhood on them. But that we are changing them in some senses. And does that change in the moment the way that we need to deal with them in the laboratory, and that that's sort of the ethical, the other stuff is sort of almost not applicable in a sense. So it's sort of question of, is it conferring humanity, even is it conferring personhood, but that you are changing the mice, and does that require changes on the ground sort of in the lab right away, and how do we ensure that those happen in a timely manner? Or how do we even know what those changes need to be, and how do we ensure that they happen in a timely manner? I think that is really, that seems to me to be the most pressing ethical question. So I was just going to say thank you for identifying that. That's great.
Well, I think with that, let's close. I think we're going to talk more, but thank you very much, Riley. And